using this technique to tell us about mechanisms underlying really important human functions.
Correlation between the induced predicted electrical field and the cerebral blood flow results we obtained for anodal and also for cathode TDCS, and these were linear correlations.
So we realized that there are many minor details that should be reported in the study to make sure that the study would be reproducible with other labs. So those items are being divided among those technological items and safety noise issues and also methodological items, as you can see here from number 1 to number 17, here in these different aspects of running a concurrent tES-fMRI.
The conductive properties, if known, for all the conductive materials should be provided.
Main approaches to mitigating RF noise and non-MRI compatible filter, right? And from there, through the waveguide, that cylindrical hole in between the control room and the scanner room.
SNR is temporal stability of the signal, and this is our main parameter of interest to ensure that the fMRI recordings are stable.
Is it stimulating at different points of the task, whether it's the initial part of the task where the participant might be becoming familiarized or where they might be learning about the task, will produce different effects to whether if you're stimulating a little bit later or if you're stimulating throughout the whole task.
Again, about 70% were highly recommending to report the experiences of TES inside the scanner.
Therefore, to assess the state of reproducibility in concurrent tES-fMRI research in context of our proposed checklist, we review concurrent tES-fMRI literature.
The only time that we removed the shield for the subject was the time that we wanted to place the electrodes and bear the cap.
Hello, everyone. I'm Ekhtiari. I'm really happy that we are back together with the 4th International Network on tES-fMRI webinars. Today, we are going to talk about concurrent transcranial electrical stimulation and fMRI. And I'm hopeful that you will enjoy about the discussions that we are going to have together. So let's start with kind of the few notes from chairs. Marom, do you want to have any kind of greetings and kind of an introductory points?
Again, I want to thank you, Hamed, for taking an outsized role and always planning these amazing programs, and just to say how nice it is to see everyone.
Great. So today we are going to have different talks. People are going to talk about different issues that we have with concurrent tES-fMRI. And the first speaker, who is going to be the opening kind of the speaker for today, is going to be Dr. Charlotte Stagg. Charlie, are you hearing us?
I am. Yes, thank you.
So yeah, the floor is yours.
Excellent, thank you. Well, thank you very much, Hamed, and everybody else for putting this together, but particularly Hamed, because yes, you're doing the heavy work here, so thank you. It's great to be here and see everybody. I mean, given that people are here, I guess this topic doesn't need much introduction, right? But I'd like to spend just a couple of minutes just, highlighting why we're here and why this is important before we get into the very detailed discussion of, how we can improve these kind of studies. So, next slide, please. So we're used to imaging techniques, and I'm presuming most people here would work with them, but of course they are entirely correlational, right? And we can make inference and we can look at differences between groups, but determining causality can be very, very hard. And really, like the rest of the world, sort of 15 years ago in Oxford, we were beginning to really, 15 years ago, we were beginning to think, how can we go beyond the correlation? How can we start to not only notice that activity in the brain area has changed in a certain condition or with a certain task, but that change is necessary for the behaviour we're seeing. And that's really where this field has been born. And it's been a long journey since then. So many of you may be familiar with studies looking at studying the effects of brain stimulation. Next slide, please. Where we do brain stimulation in the scanner and we look at the areas that are affected by it. And we can use fMRI to understand electrical stimulation. And that's really useful, right? So we can understand what we're doing and we can target our stimulation approaches better. But that's not all we can do. Next slide, please. We can use brain stimulation compared with MRI to study physiology. So we can not look at the effects of brain stimulation per se, but use brain stimulation to drive physiological processes and look at the changes. Here, this is a study from our group, but there are many others, looking at using specific frequencies of alternating current stimulation to ask a question about the importance of, in this case, 20 hertz activity in motor cortex in terms of its connectivity. And we can go further still. So next slide, please. So we can look at more complicated paradigms. So, my work, like a number of others here, is primarily interested in motor function and basic physiology, but colleagues here in Oxford have taken the ideas of combining brain stimulation and fMRI and are really using it for very complicated cognitive neuroscience questions to ask about the physiological mechanisms here about working memories. Okay, so we're taking this away from the basic physiology that this started out at and really exploiting this technique to tell us about mechanisms underlying really important human functions. Next slide, please. But those aren't the only things we can do. So fMRI is a big part of this, and it's the most widely used modality when combined with electrical stimulation, but we can do other things as well. So we can look at neurochemicals, a subject very close to my heart, with single voxel MR spectroscopy, where we look at a certain region, or, click please, magnetic resonance spectroscopy imaging, where we can start to look at neurochemicals across a variety of regions. And, next slide please, we can look at white matter connectivity. We can look at the connections within networks in terms of structure. And I think it's important to bear in mind that all of these modalities can tell us really important things, and combining them with stimulation can start to give us ideas about mechanisms, about physiology. And many of the principles we're talking about here will be equally applicable to these kind of modalities, but some won't. Okay, these have different pros and cons. We have different things that make them difficult, different things we can interpret from them. So we can take the majority of things we're seeing here, and we'll talk through those. But combining them with other imaging modalities really allows us to start to explore a whole range of really quite interesting questions. So this is great. And next slide, please. But why are we here? We've got these techniques, they're great. We can put people in a scanner, we can give them electrical stimulation, and it all works brilliantly. And to some extent, that's true, but we're putting people in large magnets and then putting electrical currents through their brains. And this is something that is difficult. It's hard to do well. And if we are going to produce results that are reproducible, that are translatable, that are intelligible, that we can, start to really understand what's happening in the brain, then we need to come up with a series of guidelines about how we should be reporting these things. We don't necessarily understand all the factors that are important as to why we might get our results. So hugely thanks to Hamid and many of the other people here, over the last couple of years, Hamid's led on putting together a consensus document, next slide please, that really sought to take people with a wide range of expertise around the world and try and understand what important information we need to give, right? So that we can make our studies reproducible. So I can read a study from another lab and think, we can do that, and vice versa. So this is a massively exciting field, and I know we've got Michael Nitsche talking in a minute about the potential of the field. There's a lot of excitement here. It's a really good point to be coming into the field. It's a really good point to be asking these kind of questions because the technology is there to do it. But we do need to take a breath, I think, and things like this are very important, and say, let's really maximize the potential of this, and let's make sure what we're doing is reproducible and is real. And so, yes, with very many thanks to everybody, and thanks to me for inviting me to be part of this. I think this is going to be an incredibly useful document, and I know we'll talk a lot about it this morning. So I hope you enjoy the session.
Thank you very much. Thank you very much, Dr. Charlie. Yeah, and the next speaker would be Dr. Michael Nitsche. And Michael, the floor is yours.
Many thanks. many thanks for inviting me here. So I'm, as you all know, I'm more TES and fMRI expert by now, but we got a new MRI now, so this might change a bit. So I will talk a bit about potentials of concurrent tES-fMRI. Will give you some, show you some new studies which I think which are interesting. Next slide, please. Okay, so as you can see here in the staircase of somehow scientific quality about the strength of evidence of brain behavioral relations, non-invasive brain stimulation and neuroimaging combination is quite of a at a high position at the staircase. And one reason is what Charlie already said, that with non-invasive brain stimulation we aim for derived causal relationships between stimulation and behavior, and here neuroimaging is very important to validate the neural basis of effects of non-invasive brain stimulation, including transcranial electrical stimulation. Next slide, please. So there are different aspects for which this combination is important. One is determination of physiological effects of TES. Another one, determination of the physiological foundation of cognitive and behavioral effects of TES, and then, of course, also monitoring of the physiological foundation of clinical effects of TES. I can't cover everything here, so I will concentrate on determination of physiological effects of TES with respect to what we can do and also some new results. Next slide, please. One interesting study which appeared last year is one of Yok and al. They did combined TDCS with both fMRI, so they looked for change of oxygen in the blood, which is coupled with neuronal activity. And they could show in their study, where they did relatively short repeated stimulation protocols, 1.5 minute stimulation repeated, and then doing both fMRI in between. So that's during stimulation, that's the right side. You can see that they did not only show relatively consistent networks, functional networks, retest reliability, but they showed also that for cathodal TDCS over the primary motor cortex, there was a reduction of both activity, and this was relatively stable over sessions. What made this study also interesting is on the left side. So the question is always, you see physiological effects and you have these modelings, and there's of course a gap between the physiological effects and modeling results. So it's important to validate what kind of electrical or magnetic fields you get. And what they've shown is that their modeled magnetic fields were relatively nicely consistent, which empirically obtained magnetic fields they obtained in the MRI scanner during TES. So that's an interesting new tool, I think, to validate computational modeling of TDCS effects. Next slide, please. So we can not only do both fMRI to look for physiological changes induced by TES, including TDCS, but another measure is arterial spin labeling, which measures cerebral blood flow. And I think the first study done in this domain was done by Charlie Stagg's group. They did dorsolateral prefrontal stimulation, and what you can see here on the left side is there was an enhancement of cerebral blood flow with anodal TDCS, so reduction with cathodal TDCS, and this was not restricted to the DLPFC, but you can see at subfigure figure C, when you have the difference between cathodal and anodal TDCS, that there's a clear enhancement by anodal TDCS over the DLPFC. You can see also on the right side that this effect lasts for some minutes after the stimulation. It's also an interesting result. And next slide, please. In our group, Asif Yamil conducted a study some years ago when we were still all in Göttingen, where he had a group of subjects who underwent different physiologial measures with TDCS under resting conditions. So we did an EEG study, we did MEPs, and we did also potent ASL measures, all in the same group of subjects. And we published last year paper where we show the ASL results. So we did 15-minute stimulation with different intensities between 0.5 and 2 milliamp and measured ASL up to 120 minutes after the end of stimulation. We did the same with motor evoked potentials. In another study, we overstimulated the primary motor cortex. Next slide, please. And what it could show was, that's now left side, the upper figure, that we got relatively linear intensity-dependent effects of TDCS and cerebral blood flow. So with higher intensity, anodal TDCS enhanced cerebral blood flow to a larger degree. With lower intensity, for cathodal TDCS, we got antagonistic results, which were always stronger with stronger stimulation. If you contrast this with TMS MEPs, so that's on the right upper side, you can see that we didn't see this clear linearity. So here, not stronger stimulation resulted in stronger effects. And you can see also for cathodal TDCS, if you look closely, you can see these non-linear effects. So we got the strongest inhibitory effect as 1 milliamp stimulation. The ASL effects were also prolonged, as in Charlie's Stagg study. So these are the lower figures. You can see with anodal TDCS, we get an enhancement of excitability lasting for up to 90 minutes after the end of stimulation. For cathodal TDCS, we got a reduction. Next slide, please. And when we compared the effects with results of modeling, we could also show that there's a nice correlation between the induced predicted electrical field and the cerebral blood flow results we obtained for anodal and also for cathodal TDCS. And these were linear correlations. Next slide. We then compared the effects of TDCS on cerebral blood flow with the effects on MEP amplitudes. And what you can see at the upper row are the associations we see for anodal TDCS. You can see that especially for the early effects, we have a relatively strong association of 30 to 40% between these parameters. With cathodal TDCS, association was a bit lower. Interesting thing, and one question might be, so that the physiological results we get might be different, might be due to partially different mechanisms. So we know that beyond the effect of TDCS on neuronal activity, we get also effects on vessels. And when you look at ASA results and MEP results, one might speculate that the linear effects we get on cerebral blood flow might be due also to stimulation polarity dependent effects on cerebral blood flow and stimulation polarity dependent effects on vessels. So next, please. Beyond these more or less regional effects of TDCS, we can also obtain functional connectivity with both fMRI, and that's a relatively early study done by Rafael Polani in our lab, so now 10 years ago, where we did 1 milliamp stimulation over the primary motor cortex. And what you can see when you did a seed-based analysis, that you get not only an activation of the primary motor cortex, but also of the motor cortical network, including premotor and parietal areas. So you can also look for alterations of functional connectivity induced by TDCS in the resting brain with fMRI. Next one, please. And a more direct measure of alterations of neuronal activity and of the contribution of neurotransmitters to these effects can be obtained with magnetic resonance spectroscopy. So we know that with TDCS we induce plasticity of glutamatergic synapses, and that GABA is also contributing. Charlie Stagg showed now also 11 years ago already that we get a polarity independent reduction of GABAergic activity in the motor cortex, whereas the effects on glutamate seem to be polarity dependent reduction with cathodal TDCS. For MRS so far, we don't see a significant modulation of glutamate with TDCS, but we know from pharmacological studies that glutamate is also critical for the induction of TDCS. One interesting new study is here, one from Heimrath and colleagues. They did the 7T-MRS study, where they have shown that with stimulation over bilateral stimulation of the auditory cortex, results in different effects. So they got an enhancement of the GABA to glutamate ratio, also here independent from stimulation polarity. So this might hint to different effects of the stimulation dependent on areas and also on stimulation electrode positions. Next slide, please. So to put things together, different protocols allow to explore tES-dependent physiological alterations at different levels. Whole brain monitoring of interventional effects is possible, including functional connectivity. We can also obtain, or we obtain with MRI, mostly indirect parameters of neuronal activity, so direct effects on vessels might also contribute. Association with more direct physiological parameters such as MEPs are present, but they are moderate. And we have some heterogeneities, of course, between studies. One reason might be methodological differences. And here, of course, to have a consensus paper will be of great value to interpret studies and also difference between studies. Last slide, please. Okay, as you can see here, we have many people in our lab to conduct these studies. Many thanks for your attention. That's it.
Great presentation, Michael. We really enjoyed it. Thank you. Thank you very much.
Thank you.
So the next talk, I'll try to give you an idea about the checklist that we have developed together during last something like 2 years of discussions and how we might be able to make a better level of transparency in methodological details on how to do concurrent tES-fMRI and how we might be able to make whatever we have in the field of fMRI TES more reproducible in terms of the methodologies and how we might be able to push forward this area of research. Like many other labs around the world, we are interested, in my lab, we are interested to see how we can modulate network level activations or connectivities with different sort of kind of stimulations like TES and transcranial alternating current stimulations with different groups of students that are interested in this area. But running these studies and then even trying to make sort of closed loop stimulation and recording and trying to see how we can continuously doing stimulation inside the scanner while we are recording the activations or the connectivities that we have inside the scanner. You can realize there are many technical details in terms of how to kind of make things in the best possible way and make sure that everything is running in the best possible way. And then you realize that reporting those technical details would definitely be critically important in terms of helping others to be able to replicate those studies. So we realize that there are many minor details that should be reported in a study to make sure that the study would be reproducible with other labs. And just kind of reviewing what people have done and what people reported, we realize there are gaps that we need to kind of fill or bridge, making sure that kind of in long term, something like 10 years from now, when we have more, still be able to replicate other studies, make sure that we are all on the same page when we talk about specific results. Even talking about things like how we can manage the level of noise inside the scanner, different labs try to kind of control for that and kind of develop different settings to make sure that we can do the stimulation with different versions of phantoms. Here we have been using watermelon and trying to see how we can measure the level of noise. Even the, kind of those technical details would definitely be important in terms of, even when we talk about noise, how we record noise, how we define the level of noise inside the scanner, and all those technical details would definitely be critical in making sure that the results would be reproducible in long term. And we always have fun running these kind of technical details and technical scans, and also kind of course, we eat the watermelons after the stimulation. But that process is going to be critically important, making sure that those technical details would definitely be reported. So we realized that there are different technological items that we need to be careful that would be reported correctly and in detail. There are some safety noise issues that are really important. Also, there are some methodological items that should be considered when we design or report a concurrent tES-fMRI to make sure that other labs would be able to replicate the results. or replicate at least the methods that we have. So we have started a discussion within the international network of tES-fMRI. And as you know, we have launched this network something like 2 years ago, and we had series of webinars, and we discussed about different issues regarding the computational head modeling, how to integrate different levels of evidence into a combination of fMRI and TES. And even we had a specific webinar discussing about the COVID-related situation and how we can run studies during COVID. And you can find them on our YouTube channel. So I'm sure that my colleagues would share a link to the YouTube channel, so you can follow the series of webinars that we have been organizing before, and they are available right now online for those who are interested. So we started the discussion among a group of colleagues within the international network of tES-fMRI. I should thank Marom for kind of his leadership, kind of helping us to be able to put all these kind of technical details together and define the pathway that we want to do in terms of making a consensus. So we started with a group of people in the steering committee, as you can see here, big names in the field and those who have been kind of doing concurrent tES-fMRI for a long time. And then meanwhile, we have done a systematic review with one of the brilliant PhD students, Bergmann, in terms of making sure that we know about all the published concurrent tES-fMRI studies in the field. We know that we have something like 200 tES-fMRI studies in the field so far, published so far, and 57 of them, they have reported a level of concurrent tES-fMRI. So we included those kind of concurrent tES-fMRI studies, making sure that we know whatever we will have done so far. And based on the people who have published kind of in these 57 papers, we started to move forward and think about, okay, how we can kind of design a Delphi consensus study, making sure that we can kind of conclude about the methodological details that are important to be reported in a concurrent tES-fMRI study. So we opened a OSF page, OSF project, and we pre-registered the methodological details that we have been thinking about and the process of the study. And also we pre-registered the analysis pipeline that we designed for the specific study. So you can find all those technical details in the OSF page. And kind of, we try to make sure that every kind of single detail that we have been doing would be transparent, available on OSF. In this process, we, as I've mentioned, that we have reviewed 57 studies, and then we invited those who have a first author or senior author position among those 57 studies. That would end up to 75 experts from labs around the world, and we also had 13 members of the steering committee. So we started with these numbers. And then we developed an initial draft of the checklist within the steering committee in terms of the items that have to be reported, and also some additional recommendations. And then we shared these kind of items and recommendations with expert panel, and also kind of again ask the steering committee members to add their thoughts on revision inside. So we had a kind of a back and forth process, making sure that people would add their comments and thoughts inside. And we received feedbacks from 49 expert panel members and also 13 steering committee members. So you can find all those kind of initial drafts of the checklist that we developed in our OSF page. So we started with these initial drafts, and then we circulated these drafts and received comments from people. And as you can see here, we had a diverse set of experts and members of the steering committee that were contributing to this process. So as soon as we ended up to have a full draft of the checklist, we shared checklist within the expert panel and the steering committee again, and asked them to rate the items and recommendations that we have. So we ended up having 17 items and 42 recommendations. And we received ratings from 45 experts and 9 members of the steering committee in the rating phase. So those items are being divided among those technological items and safety noise issues and also methodological items, as you can see here from number 1 to number 17, here in these different aspects of running a concurrent tES-fMRI. And these are the results. These are the ratings that we receive from people in the expert panel and the steering committee. As you can see here, we ask people to rate each item from extremely important to highly important to moderately important, slightly important, and not important. As you can see here, you can see them in colors right now here. And these are the items. And some of the items, as you can see here, they didn't pass the per-register threshold that we designed for the consensus study. As you can see here, the item about the amount of contact medium. And there are kind of items that they haven't passed the more stringent threshold that we designed initially. So you can see that they have been marked to make sure that we have been kind of transparently reported what we have in terms of the ratings among kind of different people in the expert panel. And it would be interesting if you go inside the technical details and the kind of the wording and kind of how these things are putting together, you realize that why there are people who might not be that much interested on some of those items. But you can see we provided all these kind of ratings available for those who are interested to see the details. And also we, as you can see here, we ask people in the expert panel steering committee to rate recommendations as yes or no, should be reported in a study with concurrent tES-fMRI or no. And you can still see there are few recommendations that they didn't reach to the 50% threshold that we have been expecting to have. Things like, as you can see here, restrictions in regulations for RF filtering methods. I mean, you can see these are not the full wording of the items that we have. You need to see the checklist and the details that we have inside the checklist. But these kind of ratings are being reported. And those items that didn't pass the per-register threshold that we had, they are being labeled, as you can see here in this checklist. Okay, we ended up having the checklist on medRxiv right now. We have 75 authors contributing to the checklist, and the checklist is right now under review in Nature Protocol. We passed the initial screening, editorial screening phase, and now it is under review process, and we will keep our fingers crossed to see how would be the results and what would be the final home for this checklist that is already available on medRxiv. And what we provided in the supplements, you can find checklist in a short form that would just include items, and also in long form which includes both items and recommendations. So you can find all those kind of two different versions in a PDF format, also in an Excel format. And the good thing about this is, I mean, in long term, we are hopeful that would help people to use them like any other kind of methodological checklist in the field to report, okay, where I have reported these items inside the manuscript and how those items could be found inside the manuscript is going to be submitted. Okay, as you can imagine, it was a really interesting journey in terms of bringing people together, 75 different authors, and receiving their feedbacks, even on the kind of the discussion part of the paper, and making sure that people would be happy with the kind of the wording that we had in the discussion and other technical details that we had inside the manuscript. It was a really interesting experience. And I highly recommend you to even see the kind of the details that people provided inside the discussion. And there are many kind of interesting points inside the discussion. And we will have speakers today talking about some of those technical details that we think that are going to be important if we want to push the field of concurrent tES-fMRI forward. And we are hopeful that we encourage others who are interested to this field. And now I can see that we have 145 participants in the webinar right now. It seems that there are many people around the world who are interested to the field of concurrent tES-fMRI. And we are hopeful that this initiative would be a starting point for further collaborations and the data sharing and protocol sharing and how we might be able to help each other in this community. I will discuss about some of these potentials at the end of the webinar, discussing about how you can contribute to these kind of international interactions. And then we are hopeful that would be just basically the first step towards that international collaborative partnership that we hope to establish. Okay, so now we have 6 speakers who are going to talk about the details that we have within the checklist and the items that we included inside the checklist, and some technical details about them. Okay, so the next speaker would be Dr. Thiel Bergmann. And Thiel, the floor is yours.
Thanks, Ahmed. So I try to be as quick as possible, talking about these many items here. Next. So it will be about the different information you should provide when publishing a paper on concurrent tES-fMRI. And I'm going to talk about manufacture of MR conditional stimulators, MR conditional electrode details, electrode positioning, MR conditional skin-electrode interface, the amount of contact medium, and electrode placement visualization. Next. The first one is pretty simple, of course. The main items you should describe is the stimulator, its brand, and its model if there are multiple ones, and specific recommendations. If possible, even provide part numbers where available, or if it's customized equipment, describe it as carefully as possible. Next. Then when it comes to the MR conditional electrode details, of course, the first thing to describe is the electrode size and shape, which can differ significantly from study to study. There are standard ones, but sometimes also you just take some scissors and cut the shape you like, right? And then you should explain specifically what it's looking like. Maybe also show a picture like you see below. The conductive properties, if known, for all the conductive materials should be provided. Next. And of course, also the position and the materials of the electrode cable connections are important, as has been shown by Saturnino et al. Modeling these aspects and showing that the position where this metal connector is actually plugged in at the electrode can strongly influence the homogeneity of the electric field in the electrode and then on the skin. So this is something that's hardly ever described, but can make a difference. Next. And you might want to note that also some of this conductive polymer or rubber electrodes are actually magnetized. This may happen during the production process, but we've realized when doing concurrent tES-MEG experiments that if you take from the same batch a few electrodes and just wave them in the MEG under the squids, you get strong distortions, and for some of them you don't get any. So this may or may not have also an impact on MRI artifacts, but this hasn't been studied yet. So it's an interesting source of artifacts to look into. Next. So then it's not only about the size and shape, but also like where those electrodes are positioned. Next one. So specifically, of course, you would need to report the positioning as precisely as possible, not only stating that one electrode was over M1, but maybe specifically how it's oriented, where the longer and shorter sides are for these rectangular electrodes, and so forth. Next. We should also distinguish between the planned and the actual electrode positioning. So we might plan very specifically using individual head modeling and E-field modeling with the rows to SIMNIPs. And it's also important then to show how those electrodes would ideally be located. Next. But then it's also the actual way of positioning them. And if you first do all the modeling and then you just stick it somewhere on the head, then everything is lost. So you might then want to use a new navigation system if you're not focusing on the EG 1020 with or without a cap, or something else like using your TMS coil to find the motor hotspot and then marking it on the scalp. So in any case, it should be reported how specifically those electrode positions have been found on the head. Okay, next. This is especially important when you have experiments with multiple sessions. So that you might want to have the same position, exactly the same orientation of those electrodes again and again. And this can be done based on EG or new navigation And yeah, it's also important to note how those electrodes are actually kept in place, what kind of headgear is used, which could be bandages, nets, sticky rings, whatever. Next. And also how electrodes and the head are actually stabilized, how they're positioned in the MR relative to the head coil, and how the maintenance of this position is ensured throughout the measurement. Okay, after electrode positioning, next. I should also focus on the skin-electrode interface. So the saline solution or conductive gel that is used need, of course, to be described in detail. Next. And it should also be reported how the actual contact between the electrode, the medium, and the scalp is ensured, because especially when there's hair, which it usually is, this is not as straightforward, and you might want to describe exactly how this contact is ensured. Next one. Also, sometimes strategies to restrict the contact medium within an electrode holder, for example, should be described when you want to avoid short circuits. And actually, better methods need to be developed. One of them you can see in the picture on the lower right is used in Hamid's group. And another approach you see on these other photographs where the gel is, or the 1020 conductive paste is placed on the participant's head in between the hair and on the electrodes, not very evenly distributed, and then stick together like a two-component glue, right, to ensure that there's really contact with all those positions on the skin. Next. And the photo, as you can see, may help to actually provide a good idea how this looks like also from below the electrode and on the skin, so that one can actually try to replicate this. If there's headgear, head straps in the way, you might want to get rid of them for the photograph. And it's really important to note that the effective electrode size and shape really depends on this actual interface. If you have a huge 5x7 centimeter sponge electrode, and it actually makes contact with the saline solution just for a few square centimeters, then that's your electrode, right? That's your effective size. So it really matters. Next. As does maybe the amount of contact medium. So that's highly ever described, and it's not always so easy to handle. You put on an electrode how much paste, in particular how much saline solution you would use with a sponge, but you should describe the details of how you tried at least to face and control this issue. But new solutions like proper electrode holders would actually be desired. Next. The impedance should be reported before, during, and after. That also helps to tell us whether there's good contact, but it doesn't substitute controlling for these contact medium parameters, because you can also get good impedance if just one part of the electrode is well connected to the scalp. Okay, next one. Yeah, note that it can of course be very difficult in practice to control this variable, given that the gel is squeezed between electrode and head, and that the saline solution just flows down the head or evaporates, but we should still try to describe it as good as possible. Next one. That's the last one. Also, the electrode placement should be visualized if possible, preferably using models from 3D rendered head surfaces, but also some dummy heads or actual photographs can help. So it sometimes tells you more than 1,000 words. And if necessary, also show multiple views on the same head, so that you actually get a good idea where those electrodes are located. Okay, I think I covered all those issues assigned to me. So next slide would just be saying thanks to my team and collaborators, and then I'm handing over to the next one so that we can stick roughly to time.
Great talk, Thiel. Thank you. Thank you very much for your contribution. Okay, the next speaker would be Dr. Shereen from City University of New York. Luke, floor is yours.
Thank you, Hamid, and thank you for organizing our 4th webinar and everybody for joining. So I'll just spend a few minutes talking about RF filter and electrical wiring pattern consideration. It's something that maybe we don't think of too often, but can be important. So why are they important, right? The answer is because they avoid imaging artifacts, and particularly RF artifacts. So what do those, go ahead, Hamid. So what do those look like? Next slide. So how to identify RF noise in your images. Typically they appear almost like any other or very many other MRI artifacts as a striped line, right? You can see this as one striped line. It's a artifact that you can see on the, can you go back up? On the left image there. Sometimes there'll be several parallel lines, or even this funky pattern like in the center where the image is probably oblique. But in any case, the artifact will appear as a line that runs along the phase encode direction. So if you see that, obviously it's not good, and you most likely have RF noise that you should try to identify and remove. So next slide. There are two main approaches to mitigating RF noise, and they differ both in how the wires are sent into the scanner room, what kind of filters they use. So as you know, the MRI scanner room is insulated from outside RF noise by this large copper Faraday shield. However, anytime you take electrically conductive wire and pass it from the control room or outside of transmitting RF energy through that wire, and that wire then acts as a broadcasting antenna. And so the panel on the left here shows how one setup where you would pass from the power source a cable that goes through a non-MRI compatible filter, right? And from there through the waveguide, that cylindrical hole in between the control room and the scanner room. And that would go into the scanner room to another filter that is MRI compatible before going to the subject. On the second approach, from the power source, you plug directly to the RF penetration panel. The filter mounts on the panel, and on the other side of the panel, the cabling goes to the subject. So they have their, you might want to use one or the other depending on your setup. Next slide, please. But there are some pros and cons for both, right? So if you're going to go through the waveguide, that might be helpful if, pragmatically speaking, if you come in with your equipment, you come out and you have to set up and remove every time. It's much easier just to send a cable through the waveguide than it is to start unhooking or hooking up electrical connections to the penetration panel. You might not be able to use the penetration panel if you don't have the accessible ports, right? Pros for the RF penetration panel is that it's grounded to the MRI room. And I think that's very important, right? And it's also a safety issue. In addition, it's less bulky, there's less components. The RF filter is smaller, there's only one of them. If you're keeping your system there, it's very helpful. Cons to the waveguide, again, unlike the penetration panel method, it's not grounded. There's more components, the filters are bigger and bulkier. It requires, cons to the RF penetration panels, you need to have an available port. You might have to puncture or do some changes to your port, your penetration panel to make it happen. And setup times can be longer if you're going to be bringing your equipment into the scanner and taking it back out. I will say also that the cabling is different. So we can go to the next slide. So just some helpful tips, right? You always want to use an RF filter. If you don't, you're most likely going to get artifacts. When possible, I would recommend, it's my personal, to run electric cables through the penetration panel. Of course, that's not always possible or convenient. You want to, once we have the cabling into the scanner room, you want to avoid any loops. You want to make sure that your cabling is as parallel as possible to the main axis of the magnet. And some experiments take that quite literally from the wire straight in the front of the bore, always parallel to the field. Other methods have much longer cabling that go around the magnet. And in that case, if you're going to go around the magnet, you have to become not parallel to the main axis at certain points. That's why in those setups, you want to have the cabling as far as possible along the walls, so that the energy sort of dissipates by the time it gets to the scanner, to the subject. You also want them to be secure, right? So for example, if you have the cables themselves create magnetic fields, create small, tiny homogeneities in the brain, right? And these magnetic field homogeneities, they're also the source of the bold signal. So you can imagine if you've got your cable maybe touching the person's arm, and they have maybe a block design where they have to push buttons, right? Every time they're pushing buttons, they're wiggling this cable. And then you're going to compare that to the rest where they're not moving their arms. You could be picking up a lost signal that's a false positive just from fluctuating the magnetic field that's produced by the wire. So keep them parallel to minimize that, and secure to really help. And then next slide, please. This sort of also connects to what Thiel was talking about. You think of the current starting at the power supply and ending at the electrode, but the current keeps going, obviously when it's going through the head, but it also goes through the paste. And as I said, Thiel mentioned before, like this is a consideration, especially I think as people are starting to, as if you're doing a high density TDCS experiment where you're getting those electrodes closer and closer together, then the paste itself becomes a substantial, footprint along with electrodes, especially if it oozes out. And so you can see here on the top row, everything's working well. You've got the minimum amount of gel and the field looks great. But if you have a lot, you can create shorts and it changes the expected what the field that you're going to distribute in the brain is. And so I think that's just an important consideration. Also imagine if you're using saline or some less viscous material, it could also, create this situation more easily, more readily. And the fact that we're imaging is great because you can see where this paste, or gel ends up. And I think that's it. You can go to the next slide. I just want to thank everybody for your attention and really looking forward to the rest of our talks.
Great talk, Luke. We really enjoyed it. There are already kind of some questions and discussion in the chat box, and I asked kind of speakers to see the questions that we have. We definitely have considered time at the end of the talks to discuss about some of those kind of experiences from different labs on how to set up these issues. And I think that there are many interesting further discussions on how to do these things in the best possible way. So the next speaker would be Dr. Axel Thielscher. Axel, are you online right now with us? Okay, Axel, the floor is yours.
Yeah, thank you. So I will shortly cover the aspects on safety and noise. So when you look into the checklist, then you will find the sub-items, and I will not cover all of them. I would really suggest that you just look into the details in the paper draft. I want to give a little bit more background information about the 3 topics which I have highlighted with these blue arrows, which is what actually MR conditional means, then a few background on safety testings, what could be done and what are caveats there, and also about noise and artifacts. Yeah, thanks. Next slide, please. So basically, what is MR conditional? We often talk about MR safety. The point is that it's really difficult to prove that you have an equipment which is safe in all possible MR environments and applications. For example, you can have a wooden spoon, and there it's probably hard to imagine that this will somehow physically interact with the MR via some physical mechanisms. So for that, I guess you can make the argument that this one is really MR safe, but other equipment, especially equipment which then has cables and conductive material, it's really difficult to make come to the conclusion that it's really safe in all different usage conditions. So that's why in the MR physics field, one talks about MR conditional, which means that it has been shown to be safe in specific usage conditions in specific MR environments. And that's really important because it means that when you leave these usage conditions or you go to another MR environment, like, for example, from 3 Tesla to a 7 Tesla, and more for inclusion, actually mostly then 2 things. First of all, that we want to encourage the manufacturers of test equipment of the equipment to clearly document which scenarios they actually tested their equipment in. That's written or not. And then once you know that, you can also ensure that the equipment which you are applying then is used within the intended specifications. For example, from one manufacturer, in the documentation, you can read that this was designed for fMRI sequences, which means that it's probably totally fine to use it for gradient echo EPIs, but when you want to go to structural imaging sequences, for example, you might want to re-evaluate the safety. Okay, thank you. Next slide, please. Okay, so safety testing, obviously, on-site can have two aims. The first one is that you just confirm really the safety, again, for the settings cleared by the test manufacturer. So this is just a confirmation test that everything is fine. And as said, you might then want to test safety when you extend this to new usage scenarios like new electrode shapes, materials, positions, MR sequences, or when you want to do different cablings in particular. It's really difficult to demonstrate that the equipment is MR conditional, and it's really needs expert knowledge, which usually is not there in the typical neuroscience field. So I would ask you to also involve just other people like MR physicists in that testing. And there are some tests mentioned in the checklist which are extremely useful. For example, temperature changes underneath the electrodes. That is a very useful point to test. But when looking or comparing that, the tests mentioned there to a formal demonstration of MR conditionality, as would be required for example, FDA approval, then this is by far not sufficient. I mentioned this here just to make the point that when you test, and it's safe, that's fine. But the main point is really that when you then again make some changes, you cannot be sure anymore that it's still safe. So please just repeat testing whenever you induce changes to your equipment. And that things can go really wrong is shown here on the right, where there was for example, a burn from an ECG pad, which was actually not designed to be used in the MRI. And this can easily happen, and things like that, I think, is probably also the largest risk for test equipment, because other types of safety issues like that it's magnetic and so on are more easily controllable. So go on, please. The good news here is that basically the sequence which we use most for imaging of brain activity is gradient echo EPI, which is a low SAR sequence, which means that the radio energy transmitted by the scanner is relatively low, and this makes it also one of the safer sequences. So I guess that this has contributed a lot to the fact that so far, tests, fMRI, there have been no reports of really tissue damage, burns, or something like that. So far, everything seems fine. And I guess when you follow these the instructions in the list, it will help that this stays like that. Okay. Next slide, please. The next thing is then obviously that when you acquire data, you want to also make sure that you don't have artifacts. And there was already a comprehensive information about RF filtering, which is a tricky thing. There are actually quite standardized testing procedures which you can follow. There, I would suggest to, for example, look at the Bergmann consortium, what they have developed for functional fMRI quality control, which is based on a standardized phantom and standardized ways of analysis. And we used that, for example, here a few years ago for RTMS in the scanner, so not tES, but TMS in the scanner, but it actually doesn't matter. You get the same kind of information out also for tests. What is shown on the left in the upper part is just the phantom scanned without the equipment and with the equipment. And what you see then, that close to the equipment, there's some signal dropout, which would then indicate that you have susceptibility artifacts. And using a phantom is really nice because it's extremely sensitive and can really nicely pick up any of these. You have a very controlled geometry also, so you can nicely pick up these effects. On the second row, that's the SFNR, how they define it, which is just the SNR, the temporal stability of the signal. And this is a main main parameter of interest to ensure that the fMRI recordings are stable, which you get. Here, actually, the equipment here scored quite well, so that the SFNR of the recordings were not really affected. And this is also something which is relatively easy to implement, so it's not difficult to set up these calculations. And I think this gives really useful information about how good the quality is. In addition, obviously, you should always then go on and check the data quality also during the fMRI analysis, so that you can be sure that also your recorded data is not affected. And here's just one example where on the right from Antal and al, where the SNR was again extracted from regions of interest, indicated as yellow and red regions there, and compared again to this to a case without the equipment and with the equipment. And this again is a nice way of ensuring that also your recorded data on the subject is not really affected. Good. Next slide, please. With that, I would like to thank you and would like to again point to the paper, which I think is a great resource and contains a lot of interesting details and informative details. I'd like to thank my co-workers and collaborators working on related topics, here in particular Frode Gregersen and Lars Hansen, with whom I'm collaborating on the safety aspects of MRCDI. Thank you.
Thank you, Axel. It was a really, really nice presentation, and thank you very much for your contribution to this endeavor. Okay, the next speaker would be, let me just bring up Dr. Lucia Lee from Imperial College London. Lucia, are you with us?
Yes, I am. Great, thanks very much. And thanks to Hamid, as always, for coordinating this and putting together, chivying us all to put together this paper. I hope it's really useful for people out there to, first of all, think more clearly about the design of their experiments and the scientific basis behind some of the choices they're making in this vast parameter space, but also to help with reproducibility, which I think is something that is very important in this field. Next slide, please. So I just want to really talk about the concept of timing, so the idea of when you do your fMRI relative to your TDCS or TES or TACT or any of the electrical simulation. And also, if you're going to do task instead of simple resting, the relative timing of that. I feel this is very non-controversial now, but it wasn't so much a few years ago, which is that brain state matters. And this is now, I think, reasonably well accepted, not only in electrical stimulation, but just last week I saw a lovely paper in brain stimulation where they had repetitive TMS and showed that it was very important what the underlying EEG frequencies were doing at the time. So essentially, when you think about, so if you look at just at the sort of top bit of the slide, when we're thinking about planning our experiments, typically you might bring your participant in, they'll go into the scanner, they'll have maybe a structural sequence, a localizer, that kind of thing, and then they'll have their fMRI, and that fMRI might be one of several ones that you're doing. You might be doing a resting, you might be doing a task, and at some point you will want to do some stimulation, and essentially you're trying to say, well, what is the effect of that stimulation on my brain function? So I've just laid out some very common things that I've seen in the literature as part of doing this paper and these reviews. So the orange arrows sort of represent variations of the pre-stimulation, and it's surprising how many people don't report where exactly they put on the electrodes and when exactly they start the stimulator. And it's only sometimes by sort of really digging or emailing people that you find out. So you can put the step, you might decide you're going to, if you've got something that's not MR conditional or MR safe, you might stimulate them before they even get in the scanner. And therefore you're going to stimulate them, do your localizer, and then do your fMRI. You might, if you've got MR conditional kit, you might sort of put the electrodes on first, take them through all of their structural scans, and then start the stimulation. And I think this is all very, very important to note, because this relates to the timing of stimulation and the extent to which that effect lasts. And I don't think that is also something we're not particularly clear of. So if you go to the bottom left corner, two really important questions currently in the field are, what is the minimum time you need to induce any kind of physiological effect? And also, how long a stimulation do you need in order to induce after effects, which may be a good thing if you're trying to do that, or may be annoying if you're trying to do repeated stimulation blocks. So that's why it's really important that you have to I feel it's really important you have to report exactly when you're doing it and whether you're taking the electrodes off in between, because that will add faff time, etc. When people talk about concurrent stimulation, you really, and I would really urge people to do little diagrams that you can see in the middle left of the screen, which shows exactly when your task is, when your rest periods are, and when your stimulation, how long that lasts relative to all of this. So when someone says simultaneous fMRI and TDCS, that could mean to some people, well, they're in the scanner, I put on the stimulation, then the stimulation stops, and then I do my task. Or it could mean I've switched it on at the same time as the fMRI, and it's going on for the same time as the MRI, or it's going to do a bit of the going to do for, say, the first part of the task, and then it's going to finish. And I think we have to be really precise about that. And the reason also is that there are several studies now which show, particularly for cognitive tasks, and I'm speaking about that because that's what I know, is that stimulating at different points of the task, whether it's the initial part of the task where the participant might be becoming familiarized or where they might be learning about the task, will produce different effects to if you're stimulating a little bit later or if you're stimulating throughout the whole task. And that's very important. And then the other thing that sort of people like to do is you have an fMRI sequence, you do the stimulation, and then you do another fMRI sequence. And again, I think it's really important to report exactly the relative gaps in between those, because again, it could be that you're doing an fMRI, you're taking them out the scanner, putting the stimulation on, and then stimulating them outside the scanner, or it could be you're stimulating them while they're still in the scanner, and it's just a gap. So that's very important. And if you do something, I think we're very familiar with the idea of doing blocks of stimulation, followed by, say, a whole run of stimulation and then waiting. But, for one of our first studies, we actually did very short blocks of stimulation within a run. And at that point, we decided to, we reported not only how long each block lasted, but we also reported the total stimulation time. And again, I think that's something also to be aware of, because we don't know the cumulative effects of stimulation yet. I think that's not something that we're very confident on, and therefore you need to be explicit and open about this. And I think I said in the paper that the bit that I wrote for this was that you have to border on the pedantic, because if you're trying to recreate someone else's study or someone's trying to recreate your study, they really need to know those details. So brain state is the thing I just want to focus on a little bit. And I'm just going to illustrate it with my paper because it was the easiest thing to find figure-wise. But there are now plenty of papers, and I've listed some of them down at the bottom, which look explicitly at brain state. And I'm talking again just about electrical stimulation, but this is also has been shown in magnetic stimulation as well. But what you're doing when you stimulate, what the brain is doing is very, very influential on what the stimulation is. So it's not really that anodal and cathodal do completely different things in networks. You can see that, but it's actually they're sort of modulating and exaggerating whatever the brain network already is. And just one final thing, say this is very important for clinical studies, because if you're thinking about doing things like rehabilitation, then timing matters. So the acutely injured brain is maybe very different for the chronically injured brain, and therefore that brain state will also be different. And the other things about brain state is consider things like task difficulty, because that will affect brain state, and familiarity. So you should be stating, did you train participants on the task before they went in, and so on and so forth. So I'll wrap up there.
Thank you very much, Lucia. It was a really, really nice presentation. Thank you. Thank you very much. The next speaker would be Dr. Daniel Keeser. Daniel, are you online?
I'm online, yes.
Good, good. The floor is yours, please.
Hi, everybody, and thanks for inviting me, Hamad. And I will just talk a little bit more on the Transcranial Electric Stimulation Experience report. And you can go to the next slide. And with their TES experience report, categories such as like comfort or drowsiness are meant. And in the ratings, the importance of main items were rated by 54 experts as either extremely important or highly important. Next. And the importance of recommendation, when there was the binary choice of yes or no, there were all, again, about 70% were highly recommending to report the experiences of TES inside the scanner. However, based on 57 articles in the literature, only about 63% of the papers do report the TES experience report. Next. So we have published in 2013 the CRQ, the Comfort Rating Questionnaire. It's based on a previous study by Nitsche and Walter Paulus, and Ulrich Peimann this time back has like a little bit adjusted the questionnaire, and we used it for all our MRI experiments, and there are questionnaires retrospectively captured during the stimulation and like after the stimulation. And in this TES experience, there are important questions regarding blinding, drowsiness, pain sensation, disturbed concentration and motion. And all these factors can influence, if you go to the next slide, multimodal MRI studies. So we have used different modalities, so from task-based fMRI to resting state to structural MRI. And if you go to the next slide, then like the blinding, the drowsiness, the pain sensation or disturbed concentration can especially have impact on their functional domains, such as like the EP sequences, the EG or the spectroscopy. And motion, as we all know, has an impact, if you go to the next slide, on all categories. This, if you go one slide further, has impact on double-blind crossover designs. So if you imagine that like only in the active condition, there is an impact on experience that is not so strongly shown in the sham condition, then this can influence the, for instance, modulation of the bulk signal. So for the conclusion, there is need to report the TES experience inside and outside the scanner. What is the best way to realize this? So we've done it in the way of the CRQ, only in a retrospective manner. But we could also think about to do it directly when the subject is inside the MRI scanner, for instance, by a visual analog scale within slider. There is impact on structural and functional MRI modalities due to motion, drowsiness, pain sensation, disturbed concentration, and blinding, just to name some of them. So far, there's no standardized international questionnaire. The question would be if the CRQ or something other questionnaire could be developed together, for instance, using OSF. Thank you.
Great talk, Daniel. Thank you. Thank you very much. Thank you very much. Okay, the next speaker would be Peyman Ghobadi-Azbari from Shahid University, Tehran. Peyman, the floor is yours.
Thanks, Ahmed. Hi, everyone. I'm Peyman Ghobadi, PhD candidate at Biomedical Engineering, and it's truly an honor for me among the great scientists who have presented today so far. I'll briefly talk about assessing the state of reproducibility and transparency in concurrent tES-fMRI research. Here you can see the Spinal concurrent tES-fMRI, or briefly ContES checklist, which, as was discussed, consists of 70 items in 3 categories. We were interested in knowing the current state of literature reporting the ContES checklist items and how the checklist would impact future studies. So after the checklist was finalized during our Delphi process, we were interested in knowing the extent to which each study reported the item that deemed significant to reported in concurrent tES-fMRI manuscript. Therefore, To assess the state of reproducibility in concurrent tES-fMRI research in context of our proposed checklist, we review concurrent tES-fMRI literature. We have used the database of one of our other studies, which was a systematic review of all the study combining tES with both online and offline fMRI, which you can see on the left hand. On the right hand, you can also Prisma flowchart of the systematic review. Please, next slide. But for specific Delphi study, we were only interested in concurrent tES-fMRI studies. So we had to recheck our database to choose our studies of the interest. To investigator independently screen the title and abstract of 568 full-text publication without reviewing any data, and excluded 268 studies based on the predefined exclusion criteria, which yield 290 studies. Subsequently, the full text of each of the 290 studies were carefully evaluated, resulting in the further exclusion of 137 studies with the reason given the Prisma flow chart. This results in 162 articles. 57 of each are concurrent tES-fMRI articles. This database had also been already used at the beginning of the process of current Delphi study to invite the expert panel. Please, next slide. Here you can see the process of the checking the state of reporting of ContES checklist among the 57 articles. For each study, 3 independent raters evaluated whether each item on the proposed checklist was presented in main manuscript or supplementarial or not. They consist of raters' response resulting in Flisch kappa of 0.95, indicating that consistency in almost perfect agreement. Inclusion of the information about the item of checklist varied widely, ranging from fully reported, such as concurrent tES-fMRI timing and imaging session timing, or rarely reported, such as the amount of contact medium. The state of reporting the ContES checklist item in a single study also varied relevantly between articles, ranging from 24% to 76%, averaging 53 of the checklist item reported in each single article. In total, our review of literature revealed a general lack of sufficient information to fully reproduce the ContES checklist. For example, over 2/3 of the study neglected to report the RF filtering method or wire routing pattern, and only have reported MR conditional electrode details. Therefore, to address this issue, we propose adapting the ContES checklist to report at least the minimum information necessary for concurrent tES-fMRI reproducibility, offering a straightforward way to balance efficiency and accuracy. This checklist is one of the first attempts to increase transparency and harmonization within the neuromodulation and neuroimaging community. We hope this checklist will encourage other initiatives to develop similar consensus checklists in other areas of neuroscience. In the end, I would like to thank Hamed, Marom, Michael Nitsche, and Axel for their valuable role in organizing this checklist from the very beginning to the end. I also want to thank my dear colleagues who helped a lot of different stage of developing this checklist. Thank you very much for your attention. I hope we get rid of COVID-19 very fast. Thank you, Ham.
Great job, Peyman. Great job. And I should thank Peyman because Peyman was playing as the facilitator of this Delphi study, putting everything together and making sure that I'm doing the right thing, and just kind of sending me 20 messages every day in the morning, kind of reminding me of 1,000 different things that we have been following at the same time for this checklist. I mean, he was doing a really, really nice job. Thank you, Payman, for your great contribution to this checklist. Okay, perfect timing. We are exactly on time, and we have two other speakers, Zainab and then Danny. They're going to talk about some other issues related to this consortium, and we will have time for further discussions and how we might be able to move forward. Zainab, the floor is yours.
Yep. Hello, everyone. And thank you, Hamid, for organizing such a nice webinar, and glad to be a part of this. In the next few minutes, I'll be sharing our own personal experience running two tES-fMRI experiments during the pandemic in New York City, which was hit really badly. So next slide, please. As any experiment, start with protocols and approval. We needed to get approval from different levels, starting with the institution, then MR facility, and making sure the subjects that are coming in do not have any symptoms or fever. So the one thing that the university imposed on us was the limitations on recruitment. So whoever that was our subject needed to be a CUNY affiliated person, either researcher or student. That was a really hard one on us. And then for the MR facility, we want to make sure everything is limiting the number of researchers that are accessing to the building, and also getting the temperature at the beginning of the entrance of the building to make sure the subject and the researchers do not have any symptoms or fever. Next slide, please. We wanted to go a little bit of an extra mile, so we removed like the paper forms of the questionnaires. So all the subject needed to fill out the Google Forms to just get rid of the paper form and traces of the virus if there was any. And all the subject and the researchers needed to have masks, shields, as well as the gloves throughout the whole time that they were inside the facility. And also our facility is really brand new, so the ventilation system was not an issue for us. The only time that we removed the shield for the subject was the time that we wanted to place the electrodes and bear the cap. We tried to be as careful as we could. Next slide, please. And then after that, the shield was back on, and we moved the subjects to the control room to make sure the impedance is okay, and we are all set to start the experiment. As you can see, the subjects are having fun with all these things. Next slide, please. Based on the guideline that MR facility had, the subject inside the scanner needed to have mask and also shield. Which at the beginning of the experiment, we started that is impossible because of the different head sizes and the head coil size. It was practically not possible for all the subjects to have the shield. So after negotiations, the shield was removed and the experiment was going on without having shield inside the scanner. And also we asked the subject, they can remove the mask when we were leaving the scanner room. And but one and only subject was allowed for 1 day, so we were making sure there's not like many subjects coming in and out. And also the scanner was cleaned immediately before and after the experiment to make sure the subjects are safe. And next slide, please. And last but not least, our MR staff and the researchers needed to be safe. So the good thing that the MR facility did for us was putting a divider. I think the last slide has this divider highlighted. That made sure we are not crossing the line. Neither the researcher nor the technician we were not using technically the same machine or the mouse or anything to make sure we are safe. And that was our own experience. So far, we are in the middle of the experiment. It's still going on. And luckily, we didn't have any issue reported by now. And obviously, this is not the best practice ever. It was the best we could do with what we had. And I hope it can help. And also at this point, I need to thank Shereen, who is here, who helped really, really much to run the protocols seamlessly, and also Marom Bikson. And that's it.
Great job, Zainab. I mean, both for your great presentation, also being able to kind of really start running kind of studies. And we had a kind of a really interesting discussion among different labs, how we might be able to be back to running studies with TMS trials. And we concluded to have a sort of a recommendation paper, and we put that together and it's published in Brain Stimulation Journal. I hope that my colleagues would share that here in the chat space. So that is something that we need to move forward with all the complexities that we have with this COVID situation. It's amazing to see kind of talented, brilliant PhD students who are pushing these kind of projects forward. So the last speaker would be Dr. Danny Wang from USC. We had a discussion with Danny in terms of how these activities within the international network of tES-fMRI could be merged with some of the activities that he is pushing forward for making a chapter in ISMRM for a combination of neuromodulation and neuroimaging. And there are great potentials for these interactions. So we decided to invite Danny and ask Danny to kind of present what he's going to do and how we might be able to contribute on that great initiative that he's putting together. And it would be a sort of an introduction for further discussion among us today to see how we can plan for the next steps in terms of presenting and being more active in the other international communities like kind of human brain mapping or ISMRM, and think about how we might be able to push this agenda forward. So, Danny, could you hear us right now?
Yes. Can you hear me?
Yes, perfectly well. The floor is yours.
Yeah. Okay. Yeah, so thanks a lot for inviting me. So I'm a newcomer to this network. So I ran MI more like a technology lab. I'm a biophysicist by training. So one of the applications is using innovative MI technology to image TES, as well as other neuromodulation techniques. Please forward. So basically, as Hamid mentioned, so we have been pushing this agenda for a while at the International Society of Magnetic Resonance in Medicine, ISMRM, which is my more like a home society. So, it intersects, I would say, with this network. Of course, majority of my work has been TDCS, but we also want to involve, include more other techniques, for example, TMS, DBS, and the emerging optical and ultrasonic kind of stimulations. So there's actually various techniques, MI techniques have been developed that you can apply concurrently or either pre and post to visualize the effects, understand the neuromechanism, and improve their efficacy. So, and we actually have scheduled, it's called a member-initiated symposium at ISMI, as well as actually OHBM this year. OHBM, ISMI was headed by me. OHBM was headed by my colleague Yi-Hong Yan. So, the topic is, I think it's timely. It's matching the BRAIN initiative by US NIH, as well as worldwide kind of government and agencies. Also, it matches, the emerging interest in brain-computer interface, such as the Neuralink. Please move forward. Yeah, so I apologize. It's very wordy, but We have actually just submit this proposal last week to IS-Mind. So this was built on our member-initiated symposium this year. So we scheduled 2.5-day workshop in fall next year. So this is almost a year and a half away. But this is just give enough buffer, for COVID to subside. And we were going to cover various techniques, including electromagnetic field mapping, functional connectivity, both arterial spin labeling, temperature, and acoustic radiation force imaging. So we'll also cover some preclinical and cellular models, as well as safety issues, as everybody is very interested, and a clinical application as biomarker development. And there will be industry partners involved. Please move forward. Yeah, so many of you guys probably have been harassed by me and probably my colleagues during the past few weeks. We have been campaigning for this workshop. We send surveys either by email and the social media. So we got a very good response. We got almost 100 positive response. Some are IS-Man members, many are not. But eventually, we organized this committee. Many of you guys are on the list. Try to, be more inclusive, to involve more junior kind of level investigators, as well as, people around the world. Please move forward. Yeah, so basically, we have just submitted proposal. Hopefully, we'll have positive response very soon. And, once we get approval, hopefully, I think we have a very good chance. Then we start planning and approach industry sponsors, and as well as many of you guys. So if you have any suggestions, volunteering, feedback, please contact me. Thank you very much.
Great, Danny. Thank you. Thank you very much for the presentation. And I think it's a really important kind of way to see how we can bring people together, not only from the tES-fMRI field, but also from other areas of neuromodulation, and see how we can help each other and push this agenda forward. So that was basically an opening to the further discussions on how we might be able to push forward this initiative that we have started, and how we might be able to move forward with the other things that we probably can do together. So we have been in touch with people within the Enigma Consortium. The Enigma Consortium is trying to put people together and kind of convince them to share their fMRI databases in different disciplines. For example, we are already collaborating with a specific Enigma working group we call Enigma Addiction Working Group. So we share our database within that consortium, and then we have kind of publications with something like 1,000 subjects from different labs. So that is the idea of Enigma Consortium, to make it possible for do some mega analysis. And we have also been talking with some other working groups. It seems that there are potentials in terms of having a specific Enigma working group on sharing data, fMRI databases that are collected during or after TES, TMS studies. So that might be one potential in terms of thinking about how we might be able to put together a consortium of labs who are interested to share databases that are collected during or after or before and after different neuromodulatory interventions, and how we might be able to kind of find common platforms for shared analysis and further discussions and replications between different labs. So that would be one of the options that we have, and we can discuss about that today. The other option would be thinking about some potentials with having a specific chapter in ISMRM, as Danny has mentioned, or having a specific special interest group within the OHPM community to be able to push this agenda forward and make sure that we are well connected, and we will also support other labs around the world to do quality research. Because at the end of the day, we are highly dependent to the kind of the results that we are making in different labs around the world. And it would be nice if we can increase the quality of research, just making sure that what we have from different labs would be kind of useful for pushing this kind of scientific field forward. The other issue would be how we might be able to have joint workshops, as Danny has mentioned, that how we might be able to have specific workshop discussing about the technical details, and also how we might be able to have next webinars. Right now, we are discussing about next webinar, talking about how we can have individually optimized brain stimulation based on EEG, fMRI, fNIRS, the different sort of brain stimulation intervention, non-invasive brain stimulation interventions. So we have already opened the floor for further discussions, not only within the tES-fMRI community, but also from other communities that are working in parallel to what we do in tES-fMRI. So we are planning for that for May 20th. It's something like 3 months from now. And we have already started discussion with Marom and a few other colleagues. We have been discussing about that with Thiel, and we have already started to kind of put together an agenda for that. So if you have a specific topic that you want to discuss about in this specific area, we definitely would be happy to receive feedbacks and suggestions from other colleagues. Please feel free to just send us an email. So I would ask my colleagues in the kind of the network to also share the Gmail account that we have, to if people are interested to kind of ask questions or kind of explore potential. We definitely would be happy to receive emails on our Gmail account, and that would be something that we can do together. So, but it is just kind of one of the some of the things that we can do together. But the floor is open. I think that Marom definitely would have ideas for that. Other chairs of the panel, Michael and Charlie, it would be nice if you open your videos and see how we can kind of extend these discussions among us. And we'll have another something like 20 minutes to be able to discuss about these issues and how we might be able to extend these collaborations.
I mean, I can say I'm thrilled for this next webinar. I know the kind of content we're starting to put together, and I'm just thrilled for the diversity and depth of speakers we're going to have. And it is including going beyond just fMRI, right? So that is an important point. And in fact, the majority of these certainly closed loop experiments are going to be EEG based. And so that is an important point for this group to consider about, you want a big tent, but you want to keep your focus as well. And so there is a balance there.
Yeah, no, I think this next webinar looks fantastic. I'm put it in my diary. I think, I've really enjoyed listening to this afternoon. I think it's been a really interesting view over a whole range of different aspects. We've had a lot of speakers talking about the very technical aspects to some of the more cognitive aspects and the sort of psychology and neuroscience, and how important all of these are, which has really highlighted, I think, the need for this kind of document. I think it's going to be, as Marom sort of suggests, I think there's going to be a challenge in taking that expertise and working out which bits apply to the combination of brain stimulation with EEG, because many of the ways we think about these things may well be just as applicable, actually, and which bits less so, and what we need to sort of consider that's different. But what are the commonalities that we can bring to this? But it's been, I've been really interested to see so many different. Just so many different aspects considered, really, and the imagination of, hearing how you can do these kind of things in the pandemic in New York. I'm absolutely amazed. We haven't, and it's given me new confidence that it's possible and that science is continuing, and that's fantastic.
Oh, just don't have too much to add. So that's just a fantastic meeting and looking at the things from different angles. And I think it will really hope the field when people are interacting more. So I'm also think quite happy about the idea to think about getting some interest groups in the larger societies, which integrates this also a bit more, and it doesn't have to be restricted to TES, of course. So could be about neuromodulation, and I think the idea is relatively old to bring stuff together. I think Vince Clark is not here today. He is promoting this also since years, And to start, this would make absolute sense. So we might have to think about the strategies there to find also people who can help to promote this. For data sharing, that's also, of course, it's also an important thing. I've a bit, so it depends on the kind of data, maybe also a bit you have. For functional data, I have a bit of doubt that at the current stage you will gain lots of it because of the problem of different protocols in each study. So we see lots of heterogeneity there, which makes it difficult to put things then really together. For structural MRI, of course, it's a different story, and if you think about modeling and so on, so that's much more standardized. So I'm all for thinking about this, and maybe establishing this will put people also at a stage that protocols are more aligned to each other between studies. But if it's useful in a very short timeline for comparing studies, I'm not so sure at the end. But it's nevertheless, it's worth the effort, I think, of course.
Yeah. No, I think Mike makes
yeah.
Very good point. Sorry, sorry, I didn't mean to talk over you. I apologize. There has been a strong presence for combination at HBM, which I guess is my sort of natural home, the Human Brain Mapping Conference, for some time, particularly spearheaded by Vince Clark, but lots of other things going on as well. The data sharing, I think, it is problematic for three reasons, one of which Michael's just very clearly delineated, which is there's just there's so much variability across studies that it's difficult to do. I think also the fact, the data is huge, right? And this is something that's a genuine issue, and what do we share and how do we make that valuable? But I think also many of the big data fMRI or MR data sharing platforms deface the data as for anonymization purposes, which then, certainly from my experience of working with Axel, it is then very difficult to do the modeling on, right? So I think there is, I think we may need to think quite carefully about whether we can use the publicly available data sets, what we might need to do to make them usable for some of the things we want to do, and the ethics around that. But I think it has to be the way of the future. We're getting very expensive, very rich data sets that should be available for everyone to test their own hypotheses and replicate the results that have been claimed.
Thank you very much. We have other speakers. May I ask other speakers to also turn on their videos and continue the discussions? And we already have lots of kind of really good questions and discussion in the chat space. So if other speakers are interested to talk about some of the questions, or people are interested to ask questions, they can post their question in the chat space, and we try to address those questions and comments that we have. And also we have people from the steering committee right now online. I can see Adam Woods, who was kind of really instrumental for us to be able to put together this checklist, and others who are already available. So it would be nice if they also contribute to discussions.
So maybe just like one point to the data sharing argument, or to the idea to share data. So we want to share, for instance, our first spectroscopy data on TDCS, and we wanted to go this road in the future. And there are some modalities where there is no need for anonymization, or where there is a strong impact on defacing and then the modeling. So meaning I could really think about on, for instance, spectroscopy for sharing, but still also like the parts of the brain, like the DLPFC gray matter, was used by us to predict or just like to find some association on better outcome in major depression. Things like this would be really interesting on a larger scale, so meaning on larger subjects. And I think it's quite promising to go into the direction of data sharing. Yes.
I'm really intrigued by this idea of current flow modeling safe defacing, right? So a defacing sequence. And I don't know, Axel, if you had more thoughts about that, about there may be a standard, there may be a way to generate something that everyone can feel comfortable about, that won't mess up current flow models.
I'm not sure I got your question fully. So is it about that the current flow influences bold activity, or what was
about developing and publicly sharing approaches to address so that the anatomical MRIs are remain anonymous.
More like a MNI. I think one would have to see how much this affects then certain electrode montages which we model, but that is all relatively straightforward to do. So I see there's no showstopper. I mean, Marom, maybe we can just look a little bit into that and design a little study or so, and then share that as maybe also as a resource how to do it, and that people can be sure that it doesn't affect too much the current flow. Yeah, that could be an interesting option.
I just want to add one more thought, and this is something that Dr. Stagg alluded to before. I think a lot of our attention has been on using imaging to figure out what the neuromodulation is doing, but there's certainly a lot of interest, obviously, in combining imaging and stimulation just to answer questions in neuroscience. I know a lot of people here have thought about it. Dr. Bergmann published a really nice review on exactly that kind of idea. So we're thinking about combining again, a lot of our attention, I think, is that there's a heavy neuromodulation focus here, and a lot of people really care as an end in itself, what brain stimulation does to the brain. But then there's a separate area of people who just want to use it as a tool to probe the brain.
Maybe following up on that, I think what a principal issue is that we sometimes have in the field where we use imaging in combination with brain stimulation, no matter whether it's TES or TMS, is that we are on the one hand still at the stage where we try to understand the mechanisms by which the brain stimulation techniques do something to the brain, and we use imaging combination with fMRI or with EEG to figure out what's happening in the brain. And on the other hand, we're already a few steps further and try to just assume that brain stimulation is doing something, and we're trying to answer, as I said, a specific neuroscientific question using those measurements, right? And it's a bit circular. I mean, of course, I'm not saying that we should wait with answering interesting questions before we've figured out all the mechanisms that would hold us back for way too long. But it's maybe good to be aware that we're doing two things at the same time, right? Trying to figure out what brain stimulation does, and already trying to use brain stimulation to have certain effects that we just take for granted when, at least when writing a proposal or an introduction, right?
And that is the background, Thiel, that could be helpful in terms of having larger databases from different labs with something like a standard protocol, making sure that we are all on the same page in terms of what is the real effect of what we do with the stimulation in the imaging data that we are collecting, and making sure that it's something replicable between different labs. And I received lots of emails from people around the world that are interested to collect data if we provide something like a shared kind of joint protocol, a standard protocol of kind of data collection in the first few steps, because they probably can do something like 10 technical scans when they are setting up the devices that they have, and share that technical scans with others, making sure that they have the basic signal that we are looking for. And they can see, for example, when you start kind of an fMRI study, you usually have something like a simple motor task, seeing do we have the activation in the motor cortex or not? Can we just do a simple finger tapping task and see what are the activations are happening there? So there are potentials in terms of if we provide a shared protocol in terms of some basic stuff that we are expecting to see when we apply the stimulation in the MRI data, that might be another opportunity to kind of share those databases together and see how we might be able to have the same results in different labs.
I think it would need a discussion on what is actually then the goal of this whole thing. So, I mean, there are so many different questions which are targeted by neuromodulation studies combining with fMRI. So what is the main goal of such a shared study? Phil can probably talk also much more about the TAX challenge, which is in a related setting, just to as a related idea, where the basic idea was really just to show very unambiguously that TAX has a clear effect on brain activity. So it was a very simple, clear question, and it's actually a lot of effort going into that. So I think the first thing we would have to do is to define a clear question which is critical to answer before we go forward, because it's a lot of work, but also interesting.
So first of all, I want to say thank you for everyone. And I have one suggestion, because I think we should think a little bit more systematically. In order to go in a jump in data sharing, we need a standard. We need a in-house code developing that different labs develop for the pre-processing and the pipelining of the images. So based on this, I think we need one more global standardization, at least to have a minimum requirement for putting the data to the shared database that everyone could use it. On the other hand, it's going to be just such a kind of numbers that no one can use it.
Marom, do you have any addition to that?
I just wanted to say that I saw that Dr. Ironside had a comment in the chat. I don't know if she wanted to just say it, and then we can comment on it.
I'm sorry I missed the first hour. I got mixed up between Central Time and Eastern Time. But if we can get buy-in from people using this going forward, it will make it so much easier to replicate and to standardize things. I was obviously engaging other academics and labs to use it is one route, but I wondered if another route would be engaging journal editors and being like, if this is something that they ask people for when they submit papers, fMRI neuromodulation papers, then it will start to become a thing, like your clinical trials registration kind of thing, where you need to have it to publish, or moving towards something like that. Is a slightly less friendly way, I suppose, of encouraging it, but another way to potentially encourage it nonetheless.
I mean, it's a good suggestion. I mean, one thing I think we want to emphasize is that the checklist, we want it to be more of a guideline than an absolute list. That you saw from the presentation of reviewing prior papers, essentially no one got it perfect, right? So including people who presented here today who are at the top of this field, you know what I mean? They weren't perfect either. And I think the fact that a prior paper didn't meet all the items on the checklist is not necessarily a deficiency, because again, it's a guidance. And so I want to be very, I think that's something we should all discuss about how, I mean, whether it's editors or frankly, the people who are here today are going to be reviewing these papers. Like, one of them is your reviewer right now. So as reviewers, there's also the opportunity to be reviewer number 2 and enforce a lot of information from the authors. And I think it is a discussion for the group of how rigid we want to be in saying you're not publishing a paper unless you literally go down the list. And I don't know the answer to that.
And I should add kind of 2 points for that, in terms of, first of all, that the paper is still under review, so we are waiting for the kind of response from the journal, but it is still citable, so people can cite it if they want to cite the kind of the technical details that we have inside the checklist. That would be helpful. And meanwhile, this is not a sort of kind of a final word in the field. So we know that it is going to be a dynamic process. So we have already started to put together a list of items that we are thinking to revise in the next version of the checklist. So Payman has already started to put something like the Google Doc, and we have already started to add some kind of notes inside. Okay, if we want to add some revisions, we might be able to change these items in the next version. And you can imagine, when you have a consensus statement, it's really hard to just go back and revise some of the items that we have concluded with some kind of like 60 votes. It's really hard to go back and revise them easily. So we need to do everything from scratch again, or at least some of this process again. But that would be possible if we can do it in something like in 2 years from now. But if you are already kind of facing with some issues in terms of using some of these items, it would be a dynamic process of sending those feedback to us. And we will try to kind of optimize some of these items in the process, and trying to make sure that this checklist would be updated in something like in 2 years from now, and making sure that it would be kind of updated frequently over the next couple of years, making sure that it would be optimized in the best possible way.
To agree with Marom that this checklist should not be implemented in the review process, like you didn't spec this one, so have a problem, but more like a guidance. And I think it's fair to say that we try to make reasonable questions, or we went through a very formal process. I think it's fair from a reviewer to ask about, okay, you don't report on that, why? And I guess it's also fair to say that when the authors then have a clear statement why they did not do something, and because they thought it's not, they should be able to give a clear answer to that, and then it should be also fine. It should be a guideline which should help to increase quality, but not a must, right?
We were also very sensitive that this document should support and not impair people going for ethics approval, that the document was written in such a way and should be understood in a way that, for example, when we talk about safety, right, there's no evidence that anybody has been hurt, and we don't see how we're operating from an abundance of caution. And so that's the other thing that we wanted to, and hopefully we got it right, that the document makes it clear, even as you go through the checklist, that this is not an absolute list, and it's ultimately the responsibility, obviously, of the PI to figure out what to manage risk benefit and figure out what is important to control in their particular study.
Charlie, do you have?
Yeah, no, I just wanted to come back to that really. That there are a number of the editors of a number of the relevant journals here, and certainly from my experience, checklists like this are very helpful in guiding, and actually it's the reviewers normally that pick them up, and exactly as Axel suggests, saying, why haven't you said this and this is a very good checkpoint. My experience of these things is that it's a lot less, it stops being something that's useful and starts being a sort of hurdle to jump over if it's compulsory, and then it just changes people's engagement with it. And actually one of the nice things about it being something that's available, that people are thinking about, that peer reviewers are commenting about, is exactly as you said, that it becomes shaped by the community, right? And it becomes clear that there are parts of this that are really valuable and really useful, and there are parts of it that possibly need looking again at, or there are things we've missed, or there are things on there that actually probably and that's a much more likely to be a collaborative and helpful and kind of evolving process if everybody signs up to it willingly rather than having it sort of mandated. So I think it's massively useful. I'll be incredibly surprised as an editor if I don't start seeing reviewers saying, I've read this and I noticed they're not conforming to, they haven't put this, and this in, why not? I can sort of already see that happening.
Thiel, do you want to add the last note?
I was just going to second what Charlie said. That it's definitely going to happen during the reviewing process. I'm seeing a lot of those manuscripts at Neuroimaging, and that's just the reviewers, and maybe also the handling editor, will just make a note. Please increase the level of detail of your methodological descriptions, and a link to the paper, which will be helpful both for the reviewers, the editors, and the authors to have a structured list, and you don't have to run on for pages saying that this and this and this detail is missing. So I think it will just happen because so many authors and reviewers are in this community that it doesn't need to be enforced. It will just happen by itself, I'm sure.
Awesome. Any other last notes from the co-chairs, Dr. Stagg, Dr. Bikson, Dr. Nitsche?
Just to say thank you very much for organising, and thank you for coming along in such a useful discussion and great talks. It's been it's been great to see. It's been really varied afternoon. I've learned a lot. So thank you, everybody, and I'm very much looking forward to the next one.
Yeah, thank you very much. This was really a great meeting, and greetings to Marom's daughter, who is hiding now.
Hello.
Hi.
Marom, do you want to say the last word, if you can hear us?
I think that the next one is really going to be amazing. We haven't quite shared the program yet, but look out for it, and it's going to be an amazing next workshop where I look forward to seeing everyone.
Great. We will keep in touch. Thank you very much, and have a nice day. Bye-bye.
Thank you.